What does a positive ANA mean? Part 1. All right, so if you get a positive ANA on your lab testing, generally the first thing we measure when we're measuring an ANA is what's called a titer and a pattern. A titer is like a number that usually reads something like 1 to 80, 1 to 160, 1 to 320. And it's not like a direct quantitative measurement. It's more of like how many ANAs are in the solution after you've diluted it a couple times. So that amount will tell you how much an ANA is present. But on top of that, what will usually be reported is what's called a pattern. Now, ANAs generally come in two types of patterns, and that pattern is based off of where in a cell the ANAs are affecting. So if you remember basic biology, this egg-looking thing is a cell. And here, every cell has a nucleus. That's the powerhouse, the brain of the cell. And then around it is the cytoplasm. That's where a lot of other things are housed, things like mitochondria. Now, if we look at the different patterns associated with ANAs, we usually have two types of patterns, a cytoplasmic pattern and a nuclear pattern. A cytoplasmic pattern are antibodies that are associated with the cytoplasm. So that's the type of the cell that is lighting up when we measure them. Okay, it's staining it. A nuclear pattern are antibodies that are going to affect the nucleus. Depending on where these antibodies are, will give us an idea of what type of conditions are potentially happening in relationship to these ANAs. So with cytoplasmic, we usually have connective tissue diseases, things like affecting the liver and the gallbladder and the muscles and things to that nature. And then nuclear antibodies tend to be more like systemic types of conditions. You're talking like lupus and scleroderma, things that can affect blood vessels in the heart and the lung and all of the tissues surrounding it. I'm going to go into more detail around what these individual patterns actually represent in terms of their individual antibodies in the next video.